My name is Matt Macauley, Assistant Professor at University of Alberta, and it's my pleasure to introduce Dr. Abhishek Bhattacherjee in my group. So Abhishek completed his PhD from University of Calcutta, where he worked on protein glycation and nanotherapeutics in the context of diabetes. He then joined the University of Alberta as a postdoc with Dr. Mirko Betty, continuing to work on glycation end products in the context of food biochemistry and microbiology. And Abby, as we call them in our group, literally walked through my door in the first year I was here, having heard that I had funding for a postdoc. And to say that that was a good decision to take Abby into my lab would be an understatement, as Abby has published a couple of really nice papers with more on the way. And he's helping to lead a highly interdisciplinary project in my lab as a research associate, and I'm going to let him tell you about that. So take it away.
Yeah, thank you, Matt, for the introduction. So I am Abhishek, and today I'm going to talk about my research that is on CD33 isoforms and how they are correlated with Alzheimer's disease susceptibility. So Alzheimer's is a global problem, and 56% of population in Canada is affected by Alzheimer's or any other sort of dementia, which simply means out of 2 of us, 1 could have a chance of Alzheimer's. So Alzheimer's is a neurodegenerative disease. Everybody knows about it. And it's basically characterized by amyloid plaque formation and neurofibrillar tangle. But in my presentation, I'm going to focus mostly on amyloid plaque formations. So in our brain, we have a protein called APP, or amyloid precursor protein, which get cleaved by beta and gamma secretase enzyme and produced monomer of A beta. Now, this is a normal process, and that's why we have a specialized macrophage cell named microglia in our brain, which can clean up this junk of monomer peptides by a process called phagocytosis and keep our brain healthy. But over time, due to several reasons, microglia is not capable to do so, and as a result, these monomers can aggregate and form amyloid beta plaque, and which is a hallmark of Alzheimer's disease. Previously, it was thought that microglia is mainly involved in phagocytosis process, but then research has shown that it actually helps on migration, cytokine secretion, and neuronal support also, which makes microglia very interesting in context of Alzheimer's disease. Now, on the other hand, genome-wide association studies has revealed that most of the genes which is strongly correlated with Alzheimer's disease, they are present in microglia, and most of them are positively correlated with Alzheimer's disease susceptibility. However, CD33 has a unique variant which 10% of the population contains, and that actually is preventive in nature, means negatively correlated with Alzheimer's susceptibility, which makes CD33 very interesting. CD33 belongs to a family of protein called Siglecs, or sialic acid binding lectins, which our lab is focused on. And there are 15 different types of Siglecs present on different types of immune cells. And I am mainly focused on CD33 or Siglec3. Siglec3 has a binding domain and a C domain, and there are 2 intercellular motifs, which is tyrosine motifs and inhibitory in nature. These motifs get phosphorylated generally, and phosphatase is recruited. As a result, they further downstream signals are dampened. This is how CD33 works as an inhibitory receptor. CD33 genetics is also very interesting. Generally, in exon 2, we have a cytosine residue, which basically are present in exon 2 in common form. And as a result, we have normal mRNA splicing and get full length of this CD33 protein, which which basically represented as long isoform or human CD33 capital M, where M stands for the major allele. But for the protective isoform, it's a little bit different. Instead of a cytosine, there is a thymine residue on the exon 2, and as a result, there is an alternative splicing. So this exon 2 get eliminated, and we get a truncated version of CD33 protein, which which is known as short isoform or human CD33 small m, which is the minor allele. Now, when we started our work, the prevailing hypothesis at that time was long isoform actually inhibits the phagocytosis process. That's why there is more A beta plaque burden, which is bad for us. And short isoform is actually a loss of function because it doesn't have the binding domain. So it don't regulate the phagocytosis process. It goes on, and there is a less plaque burden, so it's good for us. But there could be an alternative hypothesis that short isoform is actually a gain of function, and it's actually doing something to increase the phagocytosis. When we started our work, none of these hypotheses were properly tested. So the first question we asked ourselves that what's the difference between a human and mouse CD33? If there is any, then which model is good for us to study about the role of CD33? Then further, we want to understand what the long and short isoform is doing in context of microglial phagocytosis. And there is another aspect of the project where we have formulated different types of liposome engaging CD33 ligands, try to see its therapeutic benefit. But for today's presentation, I'm just going to focus on the long and short isoform and how they basically regulate the microglial phagocytosis. So as the first question I asked, what's the difference between human and mouse CD33? Is there any? So yes, human CD33 has an ITIM domain, but mouse CD33 doesn't have it. Instead of that, it has a positively charged lysine, which can couple with a DAP12 receptor and act as an ITIM or an activatory motif. To test this hypothesis, we took some mouse macrophage cell lines and mouse microglial cell lines and knock out mouse CD33 from them and tested a phagocytosis assay, a flow-based phagocytosis assay with different types of fluorescent cargoes. Now, I'd love to mention that to make our assay robust, we use different types of fluorescent cargoes, such as dextran is used to represent endocytosis process, polystyrene bead, which we used for 1 micrometer size and widely used for phagocytosis, and myelin and aggregated A beta is also pathologically very relevant, and they also represent the phagocytosis process. Now, in all of this assay, we found that the wild type and mouse CD33 knockout doesn't show any change in context of phagocytosis, which lead us to a conclusion that mouse CD33 doesn't regulate the microglial phagocytosis that strongly. And this data is also supported by a lot of animal experiments that we have done, and we have documented that in our recent publication in Communication Biology in 2019. Now, as we see, the mouse CD33 doesn't influence the phagocytosis that much. What about this human counterpart? And what about these two different isoforms? To test that hypothesis, again, we choose a human monocyte cell line, U937 cell, which is very commonly used, and we knocked out, and then genetically complement the cell by viral transduction, either with a long isoform or a short isoform or an empty vector. The empty vector basically represents the knockout cells, similar to knockout cells. And from this slide onwards, I try to keep the color coding consistent, that the long isoform is basically red in color, the data which supports the long isoform, and blue is basically for the short isoform. So when we knocked out the human CD33, we found an increase in phagocytosis. So it's very interesting because last time in the previous slide, we didn't see any difference in the mouse CD33 when we knocked out them. And we randomly choose few clones out of that and then genetically complement the long and short isoform into those knockout clones. And what we see here, the long isoform actually show a decrease in phagocytosis, which is well agreement in the hypothesis that I have shown. It's work as an inhibitory receptor. But for the short isoform, showing an increase in phagocytosis, which indicates that it is actually a gain of function. So it's very interesting for us to understand that the two different isoform is regulating phagocytosis into two different ways under in vitro condition. These lead us to the question that basically it means we need a human CD33 transgenic mice. And to do that, we basically incorporated long and short isoform genes into ROSA26 locus under the regulation of CX371 CRE. And also we tagged the GFP, so wherever this CD33 protein expresses in this transgenic mice, we can see a visual signal of GFP expression. And here are some representative slides where we can see a wild type doesn't show any signal. Actually, there are two different colors you can see in this picture. One is red, that's an IBA1 positive microglia cell, and the green is basically the GFP protein expression, and it's perfectly co-localized in both long and short isoform in the microglial cells, so that's why you can see a yellow color signal there. So it means that we made the mice properly. And further, to do a quality control, we want to understand this genetic manipulation that we have done should not influence the microglia widely. So to test that, we took different random mice from three different groups, a wild type and a long and short isoform mice. And we checked the microglial number at different regions. And we found there is no difference in the microglial number, which means this might have been properly done and this manipulation doesn't influence anything. Now it is the most interesting thing to test under this transgenic mice, what we are going to see for the long isoform. Now we have performed a competitive phagocytosis assay, and I love to explain this assay in this slide because I'm going to refer this assay into next couple of slides also. So we took a transgenic long isoform mice and wild type mice, which doesn't have any long isoform. We isolated the microglia and mixed them together and then incubate the fluorescent cargos into the same tube and then deconvolute the graph based on different immunological markers, such as CD11B positive cells represent the immune cells, whereas the CX371 and F4 positive population cells are showing a microglial population. And finally, in the flow cytometer, we deconvolute the graph based on a GFP and long isoform double positive population, which represents the transgenic wild type mice, and a double negative population, which represents the wild type mice. And we check the phagocytosis within these two populations. In this way, this assay is very well controlled and done under identical conditions. So these are all paired assay that I'm going to show from this slide. Now, as usual, we have used all the different types of cargos, and here we see a strong, significant decrease in phagocytosis in all of these cargos that we have used, which nicely fit with our result that we have shown earlier in the in vitro studies, that it basically long isoform negatively regulates microglial phagocytosis. On the contrary, the short isoform, when we perform the exact same assay, we found an increase in phagocytosis. So this in vitro and ex vivo results in microglia holds the fact that these two different isoforms are regulating the microglia phagocytosis into two different ways. One is like negatively and one is positively. So the next obvious question for us to understand, what is the genes basically upregulated in this protective isoform of the mice? To test that, we performed some single cell RNA sequencing analysis, and we took 2-month-old mice under homeostatic condition, one control, a long isoform and short isoform mice. Now, broadly, we see in the short isoform of the protective mice, there are 3 categories of genes are upregulated. One is the early response gene, which is FOSB, FOS, JUN, ERG1. Then the second category gene is KLF2, which is strongly published in the literature as an Alzheimer's protective gene. And finally, there are several cytoskeleton remodeling genes such as ROBB also upregulated in the short isoform mice. It kind of indicates the fact why this short isoform mice is showing a protective effect or like increasing the phagocytosis. But to further validate this data, we have performed RNA-scope studies with few of the gene to validate whether this higher expressions are really true or not. And we found that RNA-scope study also support the overexpression of these genes that we see in the single cell RNA sequencing. So till now, it is pretty interesting that 2 isoforms is regulating phagocytosis into 2 different ways, and we got a genetic signature that certain genes are upregulated in the protective isoform. But it's also interesting to understand what is the localization of these 2 proteins. So generally, we have a commercially available WM53 antibody, which recognizes the binding site of long isoform, so it can be easily detected. But I would love to thank GlycoNet for it because they allow us funding to develop a new antibody for the short isoform, which is unique, S503, and that basically recognizes the short isoform specifically. Now, our objective was to isolate microglia again from wild type, long and short isoform mice, and we stained them with WM53. And we see a clear signal on the cell surface for the long isoform, whereas wild type or the short isoform mice don't see any signal, which means this long isoform is present on the cell surface. But when we tested a short isoform microglia, we don't see any signal on the cell surface. However, when we permealize the cell, then we can see an increase in signal, which indicates the short isoform is actually located inside the cell. And this is also in well agreement with another paper recently published, who have shown that the short isoform is actually intercellularly located. So it is make these 2 isoforms further interesting that they are localized also differently in the cell. Finally, we want to understand all the motifs that I was talking about, ITIM and ITIM like motif, how they are involved in this process, whether they have any role or not. To test it, the experiment is very simple, where we have individually mutated either ITIM or ITIM-like residue. And we expect that if these residues are doing anything or regulating phagocytosis, we're supposed to see an opposite result. Means the long isoform generally decrease the phagocytosis, but if these residues are involved in the process, then it should increase the phagocytosis. So in the first panel, we show the equal expression of the protein. And in the second panel, we can see when we mutate the ITIM, we can see an increase in phagocytosis, which means the microglial phagocytosis that's regulated by long isoform is responsible for the ITIM residue. While for the short isoform, we exactly performed the same experiment, and we didn't see, we actually saw both of the isoform, both of the motifs are equally important here, because both of them showing a decrease, significant decrease in phagocytosis. So again, there is the difference we can see between these two isoform, where long isoform is actually regulating phagocytosis only by ITIM residue, ITIM motif, whereas both of the motifs are important for the short isoform, how it regulates the phagocytosis. Now, the next obvious question we want to understand that how this isoform is actually regulating the plaque accumulation in an in vivo Alzheimer's mice model. To test that, we took some wild type, we took some transgenic long isoform and short isoform mice and crossed them with 5x FAD mice. Now, 5x FAD mice is widely used as an aggressive mice model for the Alzheimer's disease. Now, it has its pros and cons. Like, the good thing is that it develops plaque very quickly, so it's very good for the plaque accumulation studies. Within 4 to 6 months, we can see plaque. However, the bad thing is that most of the neuronal damages might not occur that strongly, so it's not a good model to study the neuronal thing. So as we are focusing more on plaque accumulation and how these isoforms are regulating plaque accumulation, so we choose this 5x FAD model. And we want to isolate, like we want to collect the brain from different time point, 2, 4, and 8-month time point, and both male and female genders, sex, and there are at least 8 to 10 mice we want to collect for each time point. The procedure is simply basically we collect the brain, and then basically half of the brain used for microscopic studies and half of the brain used for biochemical analysis. And from this slide onwards, all the experiments are done blindly. So it's basically to make the data more unbiased. So here I will present you the 2-month-old long isoform study. Let me explain what we are seeing here. So when we cross the long isoform mice with 5x FAD at 2-month time point, we got like this 4 groups. One is the control group, which basically contains the 5x FAD gene, but no long isoform. Then that is like marked as black color. And the red one is basically the experimental group, which expresses both 5x FAD and long isoform group. And there are 3 different colors we can see in these pictures. The red one is again IBA1 positive microglia. The blue one is DAPI positive nucleus. And green one is basically the plaque accumulation or the A beta. So visually, we can clearly see there is a strong difference between the control and experimental group. And there is significant more amount of plaque accumulated in this long isoform mice, the experimental group. And that is true for both male and female. Now, if we want to recapitulate our hypothesis, we propose that long isoform is an inhibitory receptor. It inhibits phagocytosis. So there should be more plaque accumulation, and which is bad for us. And our data strongly suggests that point. So here we can see that different time points, 2, 4, and 8 months, we can see all these red lines are actually, red dots are higher than these black dots, mean the experimental group, which contains the long isoform, shows higher plaque accumulation. And that is a gradient throughout the different time. And it is true for both female and male sex. I would love to say here that each of the points actually represent one mice. So there is quite a bit of data we have added here. Now, this data is further supported by an ELISA-based study where we have quantified the soluble and insoluble A beta formation. And there also we see the similar trend as we see in the previous slide for the plaque accumulation. Actually, this red line, which is red dots, the experimental group, showing basically higher A beta accumulation, both in case of soluble and insoluble. Now, these 2 slides together proves the fact that the long isoform actually is working as an inhibitory receptor, and downregulation of the phagocytosis actually in turn increasing the plaque accumulation, which is bad for us. Now, the other interesting thing is basically the short isoform. Uh, we also have the short isoform transgenic mice, which we have crossed, but due to the experimental complexity and it's quite a long time that we need to collect the samples, we have already collected the samples and keep analyzing the data, but it's an ongoing study. So we are hoping that we see some striking protective effect in this particular group of studies. But I would but time will tell what we basically see into this thing. And now to summarize everything, I would say that the first thing we found that human and mouse CD33 are structurally different and they are different in their function also. Long isoform actually decreases the phagocytosis and it increases the plaque burden in the Alzheimer's mice model that we have shown. And the short isoform actually represents a gain of function and it increases phagocytosis, though there are further studies needed to understand how they are doing it, but it's very interesting. Now, this leads to the acknowledgement portion, and there are so different types of techniques that we have used. So I would love to thank several people who have actively collaborated with me to complete this project. First of all, Matt, obviously, I would love to thank him to allow me to work in this project. And then Chris and Emily helped to make all the cell lines and different viral transactions. Jesu actually led the project of short isoform antibody production and intercellular staining. Shushmita helped on animal genotyping. Arjun, an undergrad, worked with me, is basically helped me to do the phagocytosis assay. And there is a nice team of Gazela, Gaurav, and Vivian who basically took care about all the tissue sectioning and staining because it's the blind assay, so they didn't know. It take a little bit time to analyze the data there. I would love to thank Dr. Jason Plamel from Medicine Department of University in the Neuroscience Division and Samira. They help us to interpret the RNA sequencing data. And Mendelin from their lab also helped us to do the RNA-scope study. And Dr. Paulson and C-Tank, basically when Matt was working in the Scripps Institute, the long isoform mice was generated there, and they helped us to share the protocol so we can produce the short isoform mice in our lab. And furthermore, I would love to thank flow cytometry core facility in the University of Alberta. They helped us a lot of microglial sorting studies and high content core analysis for the RNA sequencing. And finally, the granting agencies, CHIR, NSAR, GlycoNet, and Alzheimer's Society. Thank you, and I would love to have any questions.